Hello, everyone. Welcome to the List Center. Thank you all for coming to tonight's graduate student talk. This is the final week of our Symbionts exhibition, so we're really excited to have one final program to wrap up this amazing show. Tonight, we're joined by Madeline Moore, a PhD candidate in the microbiology program here at MIT. Madeline is a synthetic biologist. And tonight she will be highlighting the importance of bio-inspired design and sharing some of her research with us. It relates to this piece right next to us by Gilberto Esparza. Madeline, take it away.
Awesome. Thanks everyone for coming, and thanks also to Elizabeth for organizing the series. It's been really fun to get to hear some of the other graduate talks, and they're also posted online if you want to check them out. I would recommend it. So I want to start off by talking a little bit about the technical setup of the piece. So it's essentially a self-sustaining system that runs off of power created by filtering wastewater samples collected from around Boston. So if you look behind me, you can see these vertically hanging columns are where the wastewater enters at the top, and it flows down using gravity. And each one of these columns contains microbial fuel cells. So these are systems that act essentially as a battery, but are powered by microbial metabolism. And so how this works is bacteria, specifically Geobacter, if you're familiar, can consume organic material like we find in the wastewater and convert it to primarily three things. So first, they make the energy that they use to power like the chemical reactions in their cells and keep themselves alive. Second, they make carbon dioxide, which they release as a byproduct. And finally, they make electrons, which they donate to the battery component of the fuel cell, and which goes on to generate electricity. So in the piece, this electricity is shuttled through these wires to create light that feeds into the core of the system, the orb in the middle. And this goes on to power the plant's photosynthesis, which creates oxygen for the animals in the core. If you look closely, you can see fish, and some that you might not be able to see that are also in there are crustaceans and single-celled organisms like protozoa. So there's a whole community of critters in there. Once the energy in the wastewater that the piece started out with is depleted, the system actually pumps the water from the core back to the top of these systems, of these columns, and the cycling continues. So I don't know about y'all, but when I look at this piece, I'm reminded initially of Earth. And what I think is so cool about this piece is that it mirrors Earth, not just visually with the orb in the center, but also in terms of what is actually going on. And that is because all of the key groups of organisms that make up ecosystems on Earth are represented and are inextricably linked by their metabolism. So we have the plants, which are acting as phototrophs that consume carbon dioxide and create oxygen. We have the animals and the microbes that are also coming in through the wastewater that are not a part of the microbial fuel cells. Those are all acting as heterotrophs and consuming the oxygen and creating carbon dioxide. And then at the core of the piece, we have microbial metabolism, which is really acting as the engine that's driving this elemental cycling. And that's similar to how the Earth is set up. Microbes play a huge role in cycling nutrients in ecosystems. So I'm going to move on to talking a bit about my research, which similarly draws from natural processes like ecology and evolution to shape life and create new technologies. So I'm sure you've all heard of natural selection. We hear about it a lot as a process, as part of the process of evolution that has played a role in shaping the diversity of life that we see today. But when you think about the organisms that we come into contact with in our daily lives, like the plants that we get our food from, or the pets that we keep in our homes, these organisms in their present form have been influenced by a type of selection that you might not consider as natural. And what I mean by this is that humans have shaped these organisms' evolutionary trajectory by choosing traits that we specifically like and intentionally breeding them to enhance these traits. So this is similar to the type of evolution that we try to implement in the lab. But but we do it on a much expedited scale, so we don't have to wait around for evolutionary time. And we do this through a process called directed evolution. And this takes place by us setting up conditions in the lab molecularly that allow us to select for biological functions that we're interested in. Yeah, and so there are a lot of ways to do directed evolution, but we in our lab specifically do this by genetically engineering organisms to link their fitness, and here that means their ability to reproduce and create offspring, to their ability to complete a biological function that we're interested in. And this allows us to select for that. So I'll give you an example. If I were trying to evolve an enzyme in our lab, let's say it is involved in a chemical conversion as part of a microbial metabolism, we would start by genetically engineering a bacteriophage, which is the organism that we use. And I also will call them phages, if you hear that later on. And these are just viruses that infect bacteria. So these are what we're genetically engineering. And we can set it up in the lab so that for the bacteriophage to be able to reproduce, it has to be carrying the sequence for a functional enzyme. And we start off, we put that sequence there with genetic engineering, and we start off with just the enzyme, our best guess at what we think the enzyme would look like that's going to be able to complete this function, just giving it a starting point. So once we have these two strains of bacteriophage and bacteria are important because the phage need a cell to infect to be able to complete their life cycle, like all viruses. We can culture these together and allow the process of evolution to take place. Luckily, we have liquid handling robots in the lab that can take care of this and help us run the experiments for weeks. I'm really thankful for this as a grad student, so I don't have to be the one staying up all night. We can pass it off to the robot. But over the course of the experiment, the phages containing the enzyme that have the best version of the enzyme will be able to reproduce more. And eventually, by the end of the experiment, they will take over the cultures. So we'll be able to collect those samples and use DNA sequencing to determine what the sequence of the most effective enzyme is that we're interested in for its function. So in this way, we're kind of setting up a microcosm in the lab where we're able to recapitulate these evolutionary principles that shape the world around us. And directed evolution has been a really successful approach so far. It's used for things that we interact with in our daily lives, for example, improving the flu vaccine or developing the enzymes in laundry detergent that we use to get rid of stains. It even went on to win the Nobel Prize in 2018. No big deal, right? But part of the reason this approach has been so valuable comes down to the vast complexity of biological systems. And this comes from the fact that in biology, we're looking at processes that break down to several scales. And we don't fully understand these processes yet. So this uncertainty kind of scales up to create a system that can contain an amount of complexity that feels kind of unwieldy and might seem messy. So directed evolution is kind of an answer to that and a way to say, okay, maybe we don't need to understand all the intricacies of the processes involved in what we want to create. We can use evolution to create this. And along the way, often what you'll find is that developing the function that you want will provide insight into the processes that we didn't understand to begin with. So that brings me to my PhD work, which I'm really focusing on how we can use evolution to shape complex systems of genes, like a metabolic pathway. And I'm interested in doing this for the obvious reason of applying it to cool problems, but also with the intention that it might allow us to elucidate kind of the design principles behind these complex systems and behind how evolution works in general. So to be able to work with these complex systems, I'm trying to implement the process of horizontal gene transfer to kind of speed up the evolution in the lab. And this is a process where bacteria can share DNA. So if you think about humans and animals, the main DNA sharing that's happening is between parent and offspring, so vertically. But bacteria are set up differently, and they can pass DNA between peers horizontally. So it'd be like if I could transfer DNA to my friend, and then that became part of their genome. And what's cool is that this DNA that they receive can actually encode information for them to be able to complete a new function. So one that we talk about often is antibiotic resistance. And this is actually becoming kind of a problem for us because it means that potentially harmful bacteria have the ability to gain genes that give them the ability to survive antibiotic treatments in humans. So that's not great. But I'm interested in incorporating this process in our experiments in the lab because I think that increasing the connectivity between the cells while we're running the experiments can actually help us reach a higher fitness. So I'll kind of walk you through how this would work. I'm early in my PhD, so it hasn't worked yet, but it's in progress. So imagine that you have a culture and 2 cells are in it. One gets a mutation in gene A that is makes the gene better. So it's a beneficial mutation. And then in another nearby cell, you get a mutation in another gene, gene B, that's also a beneficial mutation. In the absence of any kind of transfer, which the horizontal gene transfer is not a given, only some bacteria can do it, and it naturally only happens at a pretty low frequency in a culture. In the absence of any kind of transfer, theory states that one of these bacterial strains will die out and the other will take over the culture over the course of the experiment. And that's a bummer because ideally, to get the most fit metabolic pathway, we want both of those genes to be present. So if we can implement horizontal gene transfer, we can get rid of this competition and essentially have a shot at having a cell that has both of those genes in their best form. Yeah, so that's how I'm looking at improving that system. Now I want to move on to talking a bit about an application of this work, which is evolving bacterial metabolisms, which I've talked about a little bit. So for as complex as biological systems are, they're also highly specific. So for example, proteins rely heavily on their specific structural shape to be able to interact with other molecules. And this is how things happen in biology, is using structure. But that means that if a protein has evolved to recognize a certain molecule, it's unlikely that it would actually be able to recognize a different molecule and do the same thing, or even that same molecule in a different shape. So this leads into why I'm interested in creating microbial strains with unique metabolic capabilities. I'm sure some of you have taken probiotics before. I certainly have. And this is a process basically of introducing beneficial bacteria into your gut microbiome. And this makes a lot of sense. We know that your gut microbiome is really linked to your health. And so, of course, you want to supplement it by bringing in good bacteria. But research has found that it's actually tricky to get these introduced bacteria to persist in your microbiome. And the reason for that is that there's already a plethora of microbes in your system that are taking up space and other resources. So the collection of resources that an organism is utilizing is what we like to call its ecological niche. So when you introduce these bacteria, there's no available niche for them to kind of grab onto and make their living in. And so they are kind of washed away. But if we have microbial strains that can consume either rare carbon sources or synthetic carbon sources, the idea is that this would essentially carve out a synthetic niche in the microbial community, allowing these new strains to persist. And this would work likely because the existing strains in the community would most likely not be able to consume the foreign sugars, because that's a process that would have to happen over evolutionary time. So yeah, that's one application of my research. I've talked a little bit about how my research aims to draw from concepts that we see in the natural world and use them as creative forces. And I think this approach is valuable, obviously, in that it furthers our ability to create biological solutions to issues in health and sustainability. But I also like to think that drawing from concepts from the natural world keeps us a little closer to our roots on Earth as we advance technologically. And with that, I want to thank everyone for coming. I'll take any questions.